go ahead and get started. Welcome everyone to the final Wheatland seminar of the semester. My name is Tony Guay with the Wheatland Lab, and we're very excited to have Dr. Phil Townsend with us this afternoon. Dr. Townsend uses hyperspectral imaging to measure chemical functional traits and physiological status of plants, especially in response to external perturbations. He develops remote techniques to detect variations in molecular constituents in foliage, and then test those factors that cause variations between and within species and communities. Currently, this entails algorithm development for hyperspectral imagery, including from drones, airplanes, and space missions. At present, the focus of Dr. Townsend's work is on looking at continental scale variation in foliar functional traits, with work occurring across the U.S. and new work across the Arctic and boreal zones. His prior research focused on linkages between hydrology and vegetation, as well as insect disturbance impacts on ecosystem processes. Dr. Townsend is currently a distinguished visiting scientist at NASA Caltech Jet Propulsion Laboratory. He received his BA from the University of Virginia in 1989, and PhD from the University of North Carolina in 1997. He moved to the University of Wisconsin in 2005 after 7 years at the University of Maryland Center for Environmental Science. So welcome, Dr. Townsend.
All right. Thank you, everybody, for coming out. And I probably just made the mistake of drinking half a cup of coffee, so I'm probably just going to plow right into it. But everybody should, do some warm-up calisthenics here and get ready to go, because I'm gonna run through a lot of stuff, and I'm gonna try to de-jargonize some of it. So the first question I actually want to ask is, how many of you kind of have some familiarity with hyperspectral remote sensing? Feel a little bit comfortable with it? Anybody? Couple hands in there. So I'm gonna go through kind of some real basics. I'm gonna just sort of start out with some basics of hyperspectral remote sensing and what it does and why we think it can do. And then if you look at my second title right here, I'm gonna really walk through why we're using hyperspectral remote sensing to look at fine scale vegetation function, but at the continental scale. And that's where the neon comes into play. So why do I do this? Remote sensing, I think for most of us who use remote sensing, it's all about filling in gaps of information to make maps about stuff that we just can't measure everywhere on the ground. And we somehow need to extrapolate and look at what spatial patterns are, because we can't get everywhere. I'm particularly interested in the diversity and function of ecosystems, and I work in both natural and human-dominated landscapes like agriculture. But I'm going to talk almost exclusively about natural ecosystems today. So to just walk you through what I'm going to do, I'm going to talk about what imaging spectroscopy is. I'm going to talk about what functional traits are, and then what that means for ecosystem function, and then how we can use the outputs of what we're doing to understand ecosystems on Earth better. So a lot of times when you when you see imaging spectroscopy, you'll see a graphic that looks like this. This is what's called an image cube. And the idea is you've got an image, and that's just a true color image right there. If you actually look at the, it's a pointer. If you look at the spectrum right here, you can actually see the red, green, and blue wavelengths and the visible wavelengths. And this is meant to represent, there's a spectrum right there, 400 to 2500 nanometers. So this is meant to sort of show that cube dimension of all the spectral information at all these wavelengths along here that you see. So this is commonly how it's represented. And it's really, for me working with hyperspectral imagery, imaging spectroscopy is the technical term. Hyperspectral is the more widely used term, but a lot of people who are actually spectroscopists don't like hyperspectral because it sounds like it's too many wavelengths. You need all those wavelengths. But to put it in the context of sort of what we're measuring is it's really hard to convey how much information content is in hyperspectral data. So there's your visible true color. Here's a false color infrared, where you've got some random near infrared band that's shown in the red to give you a more traditional color infrared, false color image like you see on the wall over there. Or you could go into the shortwave infrared and throw that into the red band, and you get something that looks kind of like this over here. Or we can do things like the minimum noise fraction, which is a principal components analysis based transformation of the data to sort of compress all of the variation down into a smaller number of bands. At that point, you're no longer looking at particular wavelengths, but you're looking at sort of the variation. And I just randomly picked some of the principal components. So this isn't showing the top principal components or anything like that, but that's just to kind of give you an idea that it's just really hard to convey that there's 400 bands worth of data in here, and a lot of them are really highly correlated with each other, for example, in the near infrared there, but there's a lot of small variation in that we're actually interested in imaging spectroscopy. So to just kind of give you the context of what's different between multispectral data, they're showing the bandwidths for Landsat and MODIS versus what you're actually measuring. And this is a solar spectrum at the Earth, a radiance spectrum as you would see at Earth. So these are the atmospheric absorption features. This is hyperspectral, that's Landsat and MODIS. And so that's the volume of data, and that's what hyperspectral data are. So why do we do this? It's not to look for giraffes, although I love giraffes, but it's because we want to inventory our planet. And like, there's fewer than 100,000 giraffes left on the planet. And I like to use these charismatic megafauna. We pretty much know where all the giraffes are on the planet. And so for animal studies, particularly critically endangered animals like the giraffe, we actually know a lot about their distribution. But if you think about all the vegetation around the giraffe that it depends on and the ecosystems that it's associated with it, we actually know very little about those ecosystems. And remote sensing is our way to get that information, particularly in places that are not very accessible. And so for looking at ecosystem functioning, a lot of times what we do is we use flux towers, and that's a flux tower right there. And they measure eddies of gas exchange that allow us to quantify carbon uptake at sort of stand and larger scales. It's probably our best method for measuring photosynthesis at a scale that can be scaled up to the planet. And all of these different trees that are in this flux tower's footprint, much larger area depending upon wind direction and wind speed, are actually contributing to what's going on there. And it's, you know, it might be chlorophyll content, nitrogen content, all sorts of different things about that. And it leads to the question, use the Lorax here, who will speak for the trees? We know a lot through radio collars about wolves and giraffes and things like that. But how do we measure the things about trees that are important to the carbon cycle on Earth? And I would argue that hyperspectral remote sensing, among a number of other technologies, is something that we can use. So now I'm going to kind of walk you through why hyperspectral remote sensing and what it is about plants and leaves in particular that enable us to use very narrow band remote sensing to characterize them, So what is different among plants? Well, certainly there's differences in chlorophyll content, which is shown in the top figure. Then there's differences in the morphology of a leaf. So here, this is a sugar maple leaf here. You can see that there's a lot of space in here. It's a fairly light leaf. And then this is a pine needle, very compact, low leaf mass per area, high leaf mass per area. You can imagine that light interacting with these are very different. And then there's all sorts of different constituents or compounds within the tissue of foliage that have different absorption features that interact with light in a different way. So there's something that's different about plants that we're able to actually characterize and see that is based on the interaction with light. So if you think about a leaf, in this case, can anybody identify that leaf for me? Not all.
Primus. Corpus primus.
Absolutely, yeah, chestnut oak. Okay, there's a whole bunch of characteristics of this leaf that the interaction with light will enable us to detect. So here we have a typical spectrum of a leaf. Each one of these lines is a narrowband feature. The colors of the lines actually refer loosely to the different categories that I have over here. each one of these lines is associated with a narrowband feature that's associated with some sort of compound that's within the leaf, or some sort of physiological process that is associated with it, or something that's associated with the structure. So, and this is just a sampling of where some of those features are. And they're very small features that you will actually see. This is the one that's associated with phenolics right there, by the way. So there are these features at the leaf level. This is a leaf spectra that are related to everything that's going on in the ecosystem. So at the top level, there's just carbon uptake, photosynthesis, the conversion, the uptake of carbon dioxide and conversion to carbohydrates. And we're able to measure nitrogen and leaf mass per area, which are very strongly correlated with photosynthetic capacity, but also the amount of sugars and starches in there, chlorophyll and pigments, which you see mostly in these visible wavelengths, water, and then some of the macronutrients within the leaves. So this is just related to primary productivity. But there are also features, especially out here in these longer wavelengths, that are associated with the structure of the leaf that can be correlated with decomposition. In animal feed studies, they might be correlated with quality of the food, and then defense, in particular defense against insects and defense against disease. There are features in here that are associated with this that we can be able to use, that we can then be able to really quantify ecosystem function and map it. And so this is a leaf level spectra. What imaging spectroscopy or hyperspectral remote sensing is, every pixel actually has a spectrum that's associated with it that has some of these same features, plus the effects of shadow and ground and stuff like that. And you'll notice that most of these image hyperspectral spectra that I'll show you will have these areas dropped out. Those are the atmospheric absorption features for which we can't detect, which we can't make measurements through the atmosphere. So we care about this because then we can actually start to characterize what's different about the functioning of an ecosystem within a forest, between different forest types, between different vegetation types. So what are some of the applications that have come of this? I'm just going to show you some examples from my work. Certainly on a localized basis, we can do species composition, and this is actually from the Central Appalachians, and this red color right along here would be the Quercus primus or Quercus montana, depending upon where you fall taxonomically on that. But we can detect differences in species dominance. There you go, community mapping. Shout out to Dudu right here for the figure of his that I've got right here. But actually, so we can get at species, we can get at genetic variation, and actually we can get at both micro, sort of short-term genetic variation. These are aspen clones. Each one of these is an aspen clone. This is a highly stylized principal components analysis of this here. But we've actually found that hyperspectral imagery can better discriminate among clones of aspen than the chemical measurements that we make of the leaves. So aspen varies just an incredible amount chemically from clone to clone, and it's really pretty cool. But we actually can't measure all the chemistry. And the hyperspectral data is a record of the full chemistry, including the things that we can't measure. But Dudu in his work has actually shown that a lot of the spectral characteristics of different groups of plants has been conserved or not conserved evolutionarily. And so there's actually an evolutionary signal in the spectra among these different groups. And I'm not even going to try to characterize what's going on in this, but this is actually really cool and important stuff, that there's actually the spectral characteristics of a plant have an evolutionary lineage that gets passed on. And this is the basis for why high throughput phenotyping and a lot of genetic researchers are interested in using this tool as well. And then there's the expression of what's going on in the environment. This is from some work that I did in Iceland. This is a lake in Iceland that generates a lot of midges. It's Lake Mývatn. That actually means midge lake in Icelandic. The midges come out of the lake to mate. They get deposited on the land. They represent a fertilization gradient. This is actually a map of nitrogen concentration of the vegetation derived from EO1 Hyperion data. And this actually has enabled us to sort of characterize the subsidy of nitrogen from the lake onto the land and its effect on vegetation. So we have about 3+ decades now of airborne studies that have shown that we can use hyperspectral remote sensing to get at these ecosystem characteristics. It started with landmark research by Carol Westman at a place called Black Hawk Island, which is in Wisconsin. It was work done with John Aber, who subsequently went to the University of New Hampshire from the University of Wisconsin, with the predecessor to Avarice. But then there have been a whole bunch of other sort of studies that are going on. There's Greg Asner's Carnegie Airborne Observatory, NEON, which I'll talk about today. NASA has had its Avarice missions, G-Light out of Goddard, Apex, which is a European mission, and then there's this new Canadian Airborne Biodiversity Observatory. And the argument that we've been making to try and get a satellite to do this is that we want to fill in these gaps in data. And so this is showing data from 2 different studies that illustrates the problem. On both of these, you have latitude on the x-axis. So from very far south to very far north on each one of these. What this graph is talking about is essentially photosynthesis, or GPP. This is showing where the flux towers are globally. Here you go. It's in the mid-latitudes and mostly in Europe and North America. Very sparse amount of flux towers elsewhere in the world. This is actually showing where the GPP occurs on the planet. We very poorly characterize this. So if we're able to go and collect data like nitrogen LMA data that correspond very closely with photosynthetic capacity, we can fill that gap. This is a similar figure. Instead, what this is showing is how many species are present by latitude, so a lot more species in the tropics. And this is showing in a database such as TRI, which is a trait database that's widely used by people who are interested in biodiversity and foliar traits. This is showing how many of those species we actually have any trait information. And most of the trait information that we have will just be like nitrogen content or 1 trait, not even 5 or 10 traits. So once again, there's this huge gap in information that if we can actually map it, we might be able to fill that gap. And then I would argue that this is really important in the context of the just transformational global change that's going on our planet right now. So there have been a number of studies now that have shown that we can use hyperspectral remote sensing to map these foliar functional traits like nitrogen, LMA, and so forth across images. There have been studies showing it in individual locations, going back to Carol Westman's work in Wisconsin in the 1980s. But starting about 10 years ago, there started to be studies that showed that this could be done across biomes. This is a paper from Mary Martin at the University of New Hampshire showing all of these different sites. There's Howland right there, and these are all a lot of different places showing that she could build one model to predict nitrogen. My student, Aditya Singh, then actually brought that up to 7 foliar traits: fiber, lignin, cellulose, carbon, nitrogen, and LMA, and the other one is N15, or isotopic 15 nitrogen. And this is both a map, this is a map of the trait, and this is a map of the uncertainty of the trait. So we are actually mapping pixel-wise uncertainties as well. And this is across forests of the entire northeastern US, so it actually includes this area. We didn't have any images from this area, but we had some images from the Adirondacks that we used. Greg Asner is probably the best known person for doing this. He works mostly in the tropics. In the tropics, accessibility is a big issue. And in this paper here, he showed that this technology, CAO, which is what he uses, has the ability to actually map traits that we would want to know that we otherwise wouldn't be able to get into the field to be able to measure. So as an alternative, you would quantify the traits that you're interested in using airborne data rather than having tree climbers in Peru climbing up these trees and collecting all of the data. And again, to talk a little bit more about Greg Asner, probably the most effective use of this for him was he mapped all of these different traits, which are shown right here, and he's broken down how much variation is explained by them. He didn't fly his sensor over all of this area in Peru. He used geospatial methods and Landsat data to extrapolate based on a sampling that he had. But he created these sort of functional categories here based on these different chemistries. And he identified several combinations of these that were functional traits that weren't represented in the National Preserve System. And the government of Peru actually used this to create a new, very large national park. That's like a success. That's kind of the sort of thing that we're aiming towards using this data to do. We can't get into all of this place, but if we can map it, we can then use it to identify areas to target for conservation. In my own work, I'm much more focused on photosynthesis and ecosystem properties. This is a former grad student of mine. We actually found that we could use hyperspectral data through its ability to characterize traits to better characterize GPP, or total gross photosynthesis, compared to the current method that's used for MODIS. So this is the MODIS method based on what you get when you get MODIS GPP data. These are from flux towers in California. There was a California campaign of about 5 years where a lot of data were collected. All of these different ecosystem types. When we apply the SIMS model to the hyperspectral data, so this is applied to the hyperspectral data, this is not the MODIS output, because we want to compare apples and apples, you can see that we do okay, but not great, but these lines are biased from each other. Whereas when we build the model using the hyperspectral data based on traits, we get a really good one-to-one fit, and the lines aren't really that much offset from each other. So the idea is we can actually start to fill in that information about GPP as well that was on the Schimmel et al. Graph. There are a bunch of spaceborne missions coming. Right now, we just have airborne data. There's this is a free-flying satellite. These are both sensors that will be on the International Space Station. And this is a proposed sensor that NASA might launch probably by the mid-2020s. And I'm very deeply involved with that effort. These are all just sampling missions. They'll have fairly narrow swath widths. They won't cover the whole planet. This will be a Landsat-size swath width that will measure the planet every 16 days. Landsat-size pixels as well. Okay, so can we do this? So we want to map these traits across really diverse and different ecosystems. For doing this work, I'm working with NEON. For those of you who aren't familiar with NEON, it's the National Ecological Observatory Network, which is a series of 20 ecoclimatic domains. Within each one of those domains, there's about 3 sites where NEON collects all sorts of ground data, but they also have a 10 by 10 kilometer area that they fly their version of AVERIS, AVERIS being NASA's hyperspectral imager. They fly it over each one of these sites once per year. There's a few places that they don't get flown, but for example, right now they're flying over Ordway Swisher and Disney Nature Preserve in Florida, and we've got a team down there collecting foliar samples associated with this. We're using the data across all of the US as our basis to try to map foliar traits everywhere. And we want to build algorithms that would be able to work on hyperspectral data, that would work on hyperspectral data collected everywhere, and eventually moving on to all other sorts of places. I recently got funding to work in the Arctic and Boreal. I mean, NEON has a few sites in Alaska, but basically everywhere that AVERIS has flown, which are all of these yellow boxes, we want to be able to look at trait maps. Now, there's not a lot of species diversity in the Arctic Boreal zone, but there is a lot of functional diversity that's associated with hydrology, soils, global change, et cetera. NASA also had a campaign in India. It's kind of hard to see, but there are a bunch of flight boxes in India, and we've got data from the Western Ghats along the western side of India there. And then the California Hesperiid campaign, it doesn't show up very well, but over a course of 5 years, they flew they flew about a third of California seasonally every year. And the study that I showed you just a little while ago came from that data, as well as another study by Sean Serpin that looked at photosynthetic capacity, and that's in agricultural systems right there. Okay, so what do we do for this NEON study? So in NEON, it's these 80 sites that cover the variety of ecosystems that we have in North America, and that actually includes agricultural ecosystems. So basically, these images are flown And we go and identify trees within them, either using the LiDAR data, which is shown right here, using particular features. So this is actually the 1660 nanometer band depth, which is actually where we find the phenolic feature. Red maple has a lot of phenolics, so it has a very strong feature there. So this is the red maple tree that we found. So we go and we find these in these images, and we go and we sample them to actually build our model. And so this is an example of the spectra. Whenever you see this D01 through D02 through D09, that's for domain. Neon names their domains 1 through 20. And this is just giving you an idea of the spectra that came from the data. Oh. Before I go on to that slide. So we have the spectra from all of these. We have close to 2,000 samples, 2,000 plots that we have in the field. We take spectral measurements of those leaves. We dry and grind the leaves. We take spectral measurements of the dried and ground materials. We take a portion of that, and we do the chemical analysis. We do all of the measurements on it. We send them off for sugar, starches, phenolics, all of this different sort of stuff. And then we build models to predict the traits as a function of the image spectral data. There's a lot that happens in between there, and I don't think you guys want to hear about it all, but there's a lot that gets us from having taken some foliar samples on the ground and linking it to the imagery, but that's the critical part of it. It's the magic happens part of it. So we go and we build the models, and then I'm supposed to tell you a joke when I show you this slide, but these are the results. There are a whole bunch of traits here, and these are from all sorts of different functional types. We do a really, really good job of being able to map all of these traits. And this is, so for example, this is SLA, specifically area, which is the inverse of leaf mass per area. And you see that we actually build a really good model. We keep out about 1/3 of our data for validation. They never get used in the model development phase. And all this is meant to show you is that we can actually build models for most of these traits. We have a few bad outliers that you can see in here. But the really cool thing is that this works, and that this works across all ecosystem types. If we were to tune it to a given ecosystem, we would do a little bit better, but we don't have that luxury. If we want to map everything everywhere, we need to have the simplest algorithm that works across the most types of ecosystems. So this is Talladega National Forest in Alabama, which I like using because it's really pretty diverse in terms of the number of species that are there, and it's a really beautiful, beautiful forest type. And this is just showing you examples of some of the different traits for this one particular footprint. And the number right here is the uncertainty that's associated with each one of these traits. So the uncertainty for phenolics is 8.4%, which is really good. NASA has actually set, we've got a target of about 15%. We would like to have uncertainties less than 15%, so that for every pixel that we know we're within 15% of what the true value is. NASA, for operational purposes, has actually set 25% as the threshold for an operational algorithm. And actually, all of our traits have operational uncertainties of less than 25%. So we get these maps, and we have an algorithm, we have an equation that can work everywhere. So this is actually showing you the majority of the traits that we're looking at. These are validation R² and validation uncertainty right here. So this is done completely with data that's independent of the data built in the model. And you can visualize this a lot of different ways. So this is LMA in the red. So things that are redder here have higher LMA, thicker leaves. You can interpret that as conifers. Chlorophyll is in the green, and nitrogen is in the blue. So higher nitrogen would be broadleaf trees that have higher nitrogen concentration. This gives you an idea of how well we do with the independent validation data. And it's actually really cool now when you start to look at different combinations of the traits. So this one here is showing LMA, potassium, and phenolics. And you start to see some just really cool patterns. And I actually, for this particular site, I can't really explain it all that much, but you can see that there's some nuance provided to this by looking at some of these other traits. Most people who look at leaf economic spectrum type data just look at nitrogen and LMA and maybe phosphorus as well, but there's a lot of important variation on this landscape associated with other traits that we can map fairly well. And this one is one that I added after talking with Jim Clark at Duke. And what Jim Clark is interested in is masting, which is seed production in trees, and they want to use what we're doing. They're working in neon domains. They're putting out litter baskets and collecting seed crop to try to see if they can use this modeling framework, GJAM, to predict masting across all of these neon domains. And they hypothesized that the foliar traits should tell us something about it, because the allocation of resources, the outputs, the uptake and then the outputs of photosynthesis represents a decision that's being made by the tree, whether to mast, put it below ground, build wood, and so forth. And so I asked him, well, what are the traits in the leaves that would be representative of whether a tree might be masting? And he said, well, sugar, starch, potassium, and phosphorus. And I wasn't confident enough in calcium. I wasn't confident enough in my calcium model yet to put that in it. But actually, you see some just really, really cool variation here. Okay, so sugars and starches kind of make sense. It's just, I mean, some of it's a pipeline issue of just getting the sugars and starches into the main vascular system of the tree, but some of it's also a decision about resources between needing resources quickly and putting it in storage in starches. And then potassium is, I don't know why potassium is important, but it's part of one of the pathways associated with seed production. And you actually just see a lot of variation in this that you might not have seen in some of the other images. Now, I don't really know what species are here or anything like that. That would take an analysis by somebody like Jim Clark, who's actually knows a lot about masting that I don't know about to be able to do, but that's one of the potential applications of this. So once again, I wanted to just kind of convey that the big idea here is that we can map these traits everywhere. It's a single algorithm. It's the same algorithm for the grasslands at Kanza Prairie as it is in any one of these other places. We've actually completed this mapping for all of the neon domains in the eastern half of the US, including Puerto Rico, and we've done the sampling in the western half of the US, but we haven't, that was last year, and we haven't incorporated all of that data yet into it, but eventually it's going to be everywhere. And then this is our third year of field sampling, intensive field sampling, which we're going to revisit a lot of the places and visit a couple places we haven't been, to continue to validate, to make sure that this works with each subsequent year of data that we add to it. So my question for you is, do you believe this? And if I had an audience full of people who worked with hyperspectral imagery all the time, there would be skeptics in the audience. And for good reason, because a lot of this is actually based on empirical methods. And so the question is, are we actually mapping LMA, nitrogen, and phosphorus, for example, or are we actually detecting something that's really highly correlated with it? So what I'm showing you here is actually the coefficients that go into mapping each one of these. So if you look at LMA, this wavelength right here is, this indicates a wavelength that contributes a lot, and this also, so if it's highly divergent off the zero line, it's really important to the prediction. So this is LMA, this is nitrogen, this is phosphorus. Phosphorus is the reason why we would be skeptical of all of this, because there aren't any known spectral features at 400 to 25 and 100 nanometers that are associated with phosphorus compounds. So the way that we look at this is we actually look at these coefficients of each variable against each other and then see if they're correlated. And basically what you're seeing with LMA against nitrogen and LMA and nitrogen against chlorophyll, because all of these things are related to each other in terms of their function, is there's actually not any correlation. But you'll see right here that there is a pretty strong correlation between nitrogen and phosphorus. So our ability to map phosphorus is not because phosphorus has some sort of unique characteristic. It's because in the places that we've sampled, phosphorus concentrations track nitrogen concentrations. The interesting thing is that when we add Florida into the mix, where it's a phosphorus limited system, in contrast to the whole rest of the US and Alaska and everything like that, which are nitrogen limited systems, this breaks down. And that's, the Florida data is in there, but it's a very small portion of it. So this is actually just showing you the same thing. This is just, these graphs here are just asking, are the relationships between traits on a pairwise basis in our field data the same as the relationships on a pairwise basis of the traits that we derive from the imagery? And the fact that these follow along the one-to-one line, so this would be there's zero correlation between two traits. If there's zero correlation between two traits from our field data, there should be zero correlation between those two traits in the image data. and we get that. So it tells us that we're actually seeing the same relationship from our image derivations as we get from the field data. This is really important to making people believe this and actually think that this is something that they can use, like Greg Asner says, as an alternative for field mapping. I brought up to you before that every pixel has an uncertainty associated with it. We use a permutational approach to partial least squares regression for mapping. We also use Gaussian processes regression, which is a Bayesian approach, which generates uncertainties as part of it. It's a lot more computationally intensive and plus gives us the same results. So we start with permutational partial least squares regression because we have an absolutely enormous amount of data that we're working through. And so, so we can actually look at each one of the traits here and look at how many of them, what proportion of the pixels within a scene are within the 25% threshold. We can also look at what proportion of the pixels actually have predictions that are within the range of what we would expect based on the literature for what phosphorus and nitrogen and so forth, what they look like. So we can actually go and look at different combinations. So ideally, every pixel in each one of these places, right? Every pixel would have an uncertainty, would have a value and an uncertainty for each trait that is reasonable. So that would be 100% of the traits have values with a less than 25% uncertainty and are in the range of expected values. And so basically what this is showing for each one of these different domains, which line up here with these, is when you look at these, when you look at each one of these traits, what proportion of it, what proportion of the scene actually meets those requirements. And so, for example, you'll look at domain 9 and you'll see lignin, non-structural carbohydrates, so sugar plus starch, and phenolics are all fairly low. So interestingly, phenolics is one of the ones that does the best, but it does really poorly in domain 9. What is domain 9? Domain 9 is the northern plains. There's very little natural vegetation. Most of what we sampled there is like invasive plant species and non-natural vegetation. And interestingly enough, plants that don't have phenolics in it. So of course, our uncertainties are really high there because there aren't any, when you're close to zero in terms of your phenolics, your uncertainty is well outside of 25%. So we can actually sort through all of this information and figure out for each one of these domains or each one of these locations, which traits we need to be able to, which traits will give us the maximum number of pixels within a site. Because if you're doing some sort of multivariate analysis of all of these traits, you don't want to actually include pixels that have unreasonable values or have really high uncertainties because that, and so you need to be kind of careful about your interpretation. Does that make sense? Like the uncertainty analysis and the fact that we just have all this data is a way to convey to people. So our goal is to distribute this data. We want NEON to actually distribute this data and make it available to everybody so that people don't have to do this for themselves. They can go and assess how useful it is on a pixel-wise basis and on a scene-wise basis. Okay, so now I'm going to get into what we want to do with this. Are most of you familiar with the leaf economic spectrum concept? It's just this concept that came out of this paper by Wright et al. In 2004, that if you take trait data and you do a principal components analysis on it, there's basically a leaf economic spectrum. They actually found that really 3 dimensions could explain most variation in foliar function globally. And it's really from a slow return on investment. So something like conifers that have high LMA and low nitrogen, so they're investing in longevity, they have thick leaves, they're very resistant to disturbance like drought and so forth, to fast, think like aspen or something like that, which have low LMA or thin leaves, have high nitrogen content, but that tend to be deciduous or have short lifespans and have high gas exchange rates. And so this has been a way, because this has been a way that people have been sort of looking at foliar trait data to sort of explain function both within and between ecosystems. And this is from the Wright et al. Paper right here, just showing the relationships between nitrogen and mass here, respiration, and then photosynthesis, and then phosphorus. So there are these just really sort of strong relationships. It's sort of hard to show it in 3 dimensions. There are these sort of strong linear relationships. This is based on a data set. It's called the GlopNet data. It's also all in tri. It's a few hundred samples of leaves from all across the planet where they had all of these measured. This is a problem. People might have trait data, but they don't measure all of the traits everywhere. So if you go to the tri database, which is this global database, you might not have all the information. So what did we do? We went to the tri database. We wanted to see how what we're mapping across all of the US, where we have literally billions of pixels of estimates of all of these traits and all of the traits, how it compares. So this is actually our field data right here from the Eastern US. This is the relationship between nitrogen and LMA. Most of you who know about the leaf economic spectrum have seen a graph that looks like that. That's basically when you're high in nitrogen, you're low in LMA. When you're high in LMA, you're low in nitrogen. This is the relationship that we see. There are some actual interesting nuances to it though here, and that shows you the functional types that are associated with those measurements. This is what we get. So we randomly sampled from 447 of the flight lines, about 333,000 pixels. And we see, and this is a density plot, so you see that a lot falls in this range, but you see that we still have the same relationship. And I can overlay, this is the GlopNet data now overlaid on it, and you can see that we confirm that relationship, which is a good thing. We would expect to see that, but it's not, you'll notice that a lot of the data are really concentrated in a particular area, and there actually are some variations, some deviations from this that actually point to biases that people who go out and do field research have in terms of their selection of samples. And I'll talk about that in just a minute. This is that same graph that I showed earlier about where we have trait data. Again, it's all about filling those gaps. So we're now able to test what trait space looks like. Has anybody actually remember seeing a graph that looks like this from a paper or something like that? So this is actually using, again, those 330,000 pixels of all of these traits here, or it's actually 11, yeah, it's the 11 traits identified in the et al. Paper, and actually showing what, this covers 63% of the variation on all of the data, what the main drivers are. So there's a big driver here between phosphorus and nitrogen, so high phosphorus, high nitrogen, again, depending upon what your limiting nutrient is, temperate versus more tropical ecosystems, and LMA, and then some of the other things that go off of this are sort of structural components like cellulose, non-structural carbohydrates, and phenolics that are associated with other features, other ecosystem features. So we're able to actually look at this and characterize this, but now we don't have a biased sampling. We can just use all of the pixels that we have everywhere. The reason why I asked you if this looked familiar, because it looks exactly like the Diaz paper that came out in 2016, where they took 6 traits. We really only overlap with them on 3 traits, but boy, we see an almost identical looking figure. And so this is ours, this is theirs. You'll see that they have nitrogen and LMA right there. We have nitrogen and LMA right there. If you look at this here, they basically got non-woody plants, woody plants. These are our woody plants. You can't really see the colors of them. These are our herbaceous plants over here. Conifers right there. Our conifers all show up right there. These are traits that are related to seeding and wood density and things like that, which we don't have measured. We can't get that. But then these are probably actually correlated with them in some ways. So we get the same pattern out of the US data. That's like really cool. It's really fun to actually find out that we generate something that looks like a completely different data set generated. One of the things that we're really interested in is actually functional diversity. Without getting too much into functional diversity, because there's I don't know enough about how to characterize it, but one of the typical methods that is used is the convex hull method. We do this on 3 variables. Here we're just doing it on nitrogen, phosphorus, and LMA. It's basically trying to characterize functional diversity as a proxy for species diversity in these different ecosystems. We have 20 to 25 different traits that we would be able to do this on. Basically, we're calculating this. Here, what this is showing is for each of these different functional types, what the field data would show that the functional diversity is. It's actually more interesting to look at it here. So each one of these are those domains that I showed you before, different ecoclimatic regions. And this is the average functional diversity by domain, okay, based on our field data. So don't look at the y-axis. The y-axis is different because there's 330,000 pixels here. There's 1,500 field plots right here. But what's interesting is the relative position of these functional diversity, this functional diversity metric by domain. And you'll see that the relative differences is different when we look at the imagery versus when we look at our field data. This actually points to our biases in terms of what we're sampling. If we believe the imagery, we can actually see that there's some considerable differences in what the functional diversity is if we use our image data versus if we just look at the field data. So this is providing insights into what the real functional diversity might be across these different ecosystems. So it leads us to this question. Have we been asking the right questions? So people collect LMA and nitrogen data, and maybe chlorophyll, and maybe some seed size data and things like that, but they don't have the same thing everywhere. So we now can start to explore relationships that we didn't know that they were important because we now have the data to be able to do it. And I'm thinking about things like non-structural carbohydrates and phenolics, which are both really important. So the non-structural carbohydrates represent the investment in sort of short-term, and we can break this down into sugars and starches, but I've added them together for the purposes of this. But look at the investment on the least part to the products of photosynthesis. And we didn't expect to see this strong linear relationship between non-structural carbohydrates and phenolics, which are more defensive compounds. Again, pointing to some sort of adaptation that occurs there. On the other hand, with nitrogen, which is again what we typically use as a proxy for photosynthetic rate, there's no relationship. So we can actually start studying this, and we can start to break it down by ecosystem, by location, and so forth. There's just an infinite number of studies that this would actually enable. I talked to you about the biases in sampling, and I want to kind of get back to that right now. So this is, again, the typical leaf economic spectrum relationship of LMA and nitrogen, and this is our field data. You can barely see it here, but in the pink right here are actually those 330,000 randomly selected pixels. And you remember it had sort of a broad width. It's a little bit more dense right in there, but there are these areas here, right in there, that aren't represented by any of our field data. And so we can actually select that out and actually map the areas of the leaf economic spectrum that aren't in the field data and ask, is this even reasonable? And so we've done this with a lot of different combinations. This is again from Talladega. So first of all, if this were all speckled, we'd say, oh, it's just random error. Those are just random pixels. They're mixed pixels. You're seeing soil signal or whatever. But no, this is actually coming out to something coherent. And these blue patches are young conifers that have high LMA, like conifers do, but they also, as young conifers do, have high nitrogen content. I remember when I was doing my PhD with Pete at the University of North Carolina, and we would go out to set up our field plots. He would look for the most beautiful examples of an ecosystem, and we would set up our plots and make our measurements there. And then when I started doing my, after I finished and I started working in disturbed forests, we would go and look for the lousiest examples of ecosystems. But you know what? The ecologists aren't walking out into these young conifer regenerating conifers, collecting samples and putting their data in the TRI database. And I can give you, a half dozen examples of that from Neon, where we find coherent patterns of traits that don't show up in any database because there's actually a bias against sampling that, because that's not what ecologists are really actually interested in. So we're not the first people. I have to keep giving credit to Greg Asner because he's done some of the same stuff with his work in Peru. And so Interestingly enough, in this paper that was in PNAS, he actually didn't talk about the fact that his relationships actually didn't match what's in the literature. So he found these are showing his relationships too, but he didn't actually dwell at all in the paper about how this actually differs from what might be expected. And I'm really interested in pursuing that. There have been a couple papers. People are really interested in functional traits globally. And so there have been groups that have been using the TRI database and linking it to climatic and soil variables to try to predict global patterns of different traits. So this is from this paper by Ethan Butler from University of Minnesota that just uses TRI database and climate and soil and topography variables, and that's nitrogen, phosphorus, and element or SLA. In this case, these are really quite big pixels, I think maybe 50 kilometer pixels or something like that. But the sort of thing that we're doing at a fine spatial scale can actually help provide us with some detail on what's going on inside of these. And then there was this paper by Moreno Martinez, is essentially doing the same thing, again, working with the TRI database. The one thing that she did that's a little bit different is she actually used remote sensing data as well. So her pixel size is more like 250 meters, which is what the MODIS pixel size is. But now we have fine-grained validation data that we would be able to go in and look at what's going in there. And actually, we might be able to help these folks do a better job. And that's Peter Reich, who's the PI on the Butler paper that I just showed, and I are trying to figure out now how we can incorporate what we're doing. And when there's a spaceborne satellite, something like NEON or these airborne campaigns would be crucial to validation. What's cool about the Moreno Martinez paper is that she actually, in her analysis, she actually looked at what there are two bars here. One bar is actually the distribution of values. The other bar is actually the distribution of how many samples there are by different latitudes. So this is like that previous graph, except for that what was a latitude on the x-axis is now on the y-axis. And so she actually is giving an example of how they've actually filled in the gaps. Okay, so added late, because I know LiDAR is a big thing here in Maine. We want to actually integrate this with other types of data. And I haven't talked at all about the stuff with thermal. Photosynthesis is a chemical reaction, temperature is critical to it. And so if we have thermal infrared data, we can actually derive rates based on what we're doing. But actually, functional traits vary vertically within a canopy. LMA would be the most obvious one, with thickest leaves at top and thinnest leaves at the bottom. It's just an adaptation to harvesting light and the environment that the plant's in. Most of the work goes on at the top of the canopy, but this actually would allow us to get at full canopy types of work. So this is the work of my PhD student, Adam Kloos, who's interested in taking the hyperspectral data, which we use to map top of canopy traits. That's what those maps of and everything I've shown you are. And then using LiDAR to get profiles through the canopy and field data where we've collected leaf samples from within a canopy to then actually estimate the trait as sort of a voxel in three dimensions. So we have our map of the trait, we have the LiDAR, which we get sort of LiDAR transmittance through the canopy, and then we have an estimate of what the trait would look like. And this is actually, so he's got LiDAR transmittance is his estimate of the sort of density position within the canopy. So this is field data. And these are different species, by the way. This is LMA by height. You'll see that, I mean, there is a relationship. LMA declines the closer to the surface that you get. And then if we convert LiDAR to LiDAR transmittance, so the percent of the LiDAR signal that gets through the canopy at a certain height, you see that there's actually a really strong and consistent relationship among species. And Adam has used that as a basis then to actually measure LMA with height. So imagine a transect going from aspen, shorter aspen to taller maple, back to shorter aspen. You can see the height profile right there that comes out of the LiDAR data, and then using a model that he develops of how LiDAR, how traits change within a canopy, you'll see that it's actually different between the maple and the aspen right here. We can actually build a model of foliar traits with depth in the canopy by integrating LiDAR and hyperspectral derived traits. So finishing this up now. Catherine asked me, what are the challenges we face? Why isn't everybody doing this? Hyperspectral data are hard to work with. There's a lot of things that are just not obvious to people how to do. The data that gets distributed to you by the producers of the data, if you start working with that without doing anything to it, you'll make good maps of the artifacts that are in the data. And that's the real problem. The tools just aren't out there, and a lot of it's because not many people work with the data. There's been a lot of funding to work with the data. There's not a satellite, and there's not a lot of people that have been training to do it. And then the community needs tools that are open source and available to them. My workflow that has generated everything that I show there is open source. If you look at, if you search on HiTools at GitHub, you'll find it. And it's constantly being updated, but a lot of groups are now starting to try to make these tools available. And then there's other pernicious issues with how you link ground data to the image data. There's mixed pixels in the imagery that when you've got soil, it's sort of baked into these models, and I can talk to you about how PLS works and deals with that. How you scale up from the ground to from the canopy to an imagery. A lot of this, like as a community of people who do what I need to do, we actually need to kind of standardize how we do it. But there are issues that are out there, but there's work by myself, by Greg Asner, by Scott Ollinger, and so forth. They're showing that it looks like we could do this. And I'm really optimistic that if we get a global satellite, we'll be able to end up making maps of all of these traits. In my case, using a single algorithm, but maybe if you want to do really well in one of these places, it's an algorithm that's tuned to that particular location. So we've come a long way. This is Black Hawk Island, which is an island in the middle of the Wisconsin River. It's where the first hyperspectral study was done by Carol Westman of vegetation, done in the 1980s. NASA has flown Avarice over this probably 15 or 20 times. The most recent time being in 2015. And we have a hyperspectral sensor. It's not as good as NASA's, of course, but we've actually flown over it the past 3 years as well. And we'll fly it over again in 2019 as well. So we've actually got 30 years of imagery. And the last chapter of Adam's dissertation, my student who I mentioned before, is actually going to be looking at 30 years of change of foliar traits on this island as this forest has matured. There have been disturbances on this forest. And if you don't know Black Hawk Island, it's actually really famous. It's where some of the earliest and best studies of ecosystem processes linking net primary productivity and GPP to soil characteristics done by John Aber and Jerry Melillo and John Pastor and a few other people in the late 1970s and early 1980s. It's sort of the landmark soil ecosystem processes to foliar processes study was actually done there. So it's really cool. And this is probably one of the only places, maybe Harvard Forest being the other one, quite possibly the White Mountains sites of John Aber's in New Hampshire as well. But we actually have some places where we can actually look at how ecosystem functioning has changed over this time period where we know climate has changed so much. And I hope that was interesting to you. So thank you.
Questions for Dr. Townsend?
I ran through like a lot of stuff, and Tony will know that I was like busy. Like I just have this thing based on my conversations with people while I've been here. I was like adding new slides there today, and it's like, oh, what I took out a couple of slides. I took out the slide that asked the question, why are we wrong in some places? So I didn't. I do have a slide that talks about why we're wrong in some places. Yeah.
Sorry, I'll just reveal my own biases, but the non-vascular vegetation in places which we talked about yesterday has a whole suite of traits and pigments specifically that I think this type of stuff makes a lot of sense to try to use to map.
You don't have as much structure as an issue.
Right.
Yeah.
But I'm thinking also, globally, a lot of the water too. Like, is like this kind of the first step towards resolving lots of different chemical attributes, not just on land, but also not just vascular plants. I mean, is that are we far behind in those other areas?
No. So the aquatic community doesn't have its act together quite as well as the vegetation community does. But that's a function of two things. There are fewer people doing this in the vegetation community, and we tend to be a lot tighter, whereas the aquatic community has actually been doing this sort of stuff for much longer and has a very large community of people with very, very strong opinions on it. But they're about to get their own satellite, PACE, that is intended to do that. And the thing about the aquatic community is that water moves. And the traits are highly variable and highly, like, think of phytoplankton blooms or so forth. One of the reasons why I wanted to talk to you is because I don't know, I mean, I know of some very isolated case studies where people have been looking at spectra with bryophytes and lichens and so forth, but actually this kind of scale of doing that hasn't been done. And I know, starting to work in the above domain, I'm going to have to be thinking about it. And that's one thing that I need to learn a little bit more about it. And what does a trait mean when a pixel is 50% lichen and 50% some shrub or something like that as well. So there's not much that's been done there. The aquatic community for aquatic traits, of course, their traits are completely different. It's things like CDOM and particulate matter, all of these different, sorts of labile materials. They actually are in pretty good shape in terms of a lot of people have been doing it, but the big issue for them for a mission like this is that they might need more rapid return time to actually see the dynamics they're interested in. I don't know if I answered your question.
You did.
Yeah.
Other questions for Bill? All right. Well, we're gonna. Another question.
Well, I have a non-spectral person because you mentioned earlier that that there's a lack of data coming from the tropics.
Yeah.
And how is the stuff that you're doing here in the temperate region able to inform what's going on in the tropics?
So it doesn't. Okay. And I have worked in India in the tropics and in Florida, which is sort of the subtropics, and Puerto Rico. Greg Asner works exclusively in the tropics. And my big challenge and the thing that I want to do is actually integrate the data that I've been collected with what Asner has done, because I don't feel like there's any need to replicate that because he's characterized that so well. It's a bit of a challenge to do that because it means bringing people together who are busy and so forth. But I kind of feel like with the data, so they're not shown here, but I have a whole bunch of figures where Greg has given me his spectra and his models, and I overlaid them with my models, and they're the same. The coefficients are a little bit different in terms of magnitude and everything like that because he processes his imagery differently from how I process my imagery. And at some point, he and I are going to get together and we're going to mutually analyze each other's data so that we can align it.
That's exciting.
Yeah.
Any other questions? Okay, please join us at the Black Bear Brewery and Tap Room, or Tim's, probably around 5, I would say. We'll be over there. So join us if you can, and thanks for coming. We'll see you in the fall. And thanks again, Phil.
Yeah, thank you.